Welcome dear students. In this class we are going to learn about a very important phenomena that occurs during the cell cycle. That is separation of two sister chromatids during anaphase. How this process occurs and what is the role of MCDK in this process I will discuss in more details in this class. But before you start to learn this process, you must remember a protein called cohesin. You have to understand the function of this protein at first. Cohesin. This cohesin protein is actually multi-subunit protein. This cohesin protein have SMC1, SMC3 subunit. SMC means structural maintenance chromosome 1 and 3. There is SCC1 and SCC3, two other subunit. There is PDS5, WAP1 and also another subunit called soronin. So you can see here the cohesin molecule is actually multi-subunit molecules. Let me draw a cohesin molecule. If this is the SMC protein, then it is actually coil structure and has a ATPase domain, globular ATPase domain. This is the ATPase domain. ATPase domain. This is globular in structure. And let's suppose this is the, so it is SMC1, let's suppose, and here the other subunit, this is the another ATPase domain, this is called, name it SMC3, SMC3. Then two other subunit like SCC1 bind with this molecule called SCC1. Then there is another subunit called SCC3, SCC3. There is two other subunit like, let's suppose this is the PDS5 and this is the WAP1, and there is another subunit, this is soronin. Soronin. Now you can see this is a multimolecular multi-subunit protein. So what is the significance of this protein? I will show you. At first, in the G1 phase, there is chromosome. Yes, this chromosome actually attached with this cohesin molecule during the G1 phase with the help of two molecules called SCC2 and SCC4. These two molecules helps in the bringing of cohesin molecules. They actually recruit this cohesin molecule to this chromosome. So the chromosome is attached with this cohesin, but you can see the cohesin is not actually grab the chromosome, it is actually associated outside the chromosome. Okay, this is all cohesin. Another fact is other molecules are present that actually can reverse the process, that actually remove these cohesins. The proteins are called PDS5 and WAP1 that is associated with this molecule. So this process can be interchanged in the G1 phase. This is not actually, this cohesin has not now cohesive activity, but in the S phase it gains the cohesive activity. In that phase you can see here the PDS1 and PDS5 and WAP1 is present, so the reverse, this can be reversed. But during the S phase a very important phenomenon occurs, that is an enzyme gets activated called COAT. Enzyme gets activated called COAT. COAT full form is cohesin acetyl transferase. That means this enzyme can transfer acetyl group and particularly to the SMC3 subunit. So it adds acetyl group to the SMC3 subunit. And another thing is, when this molecule is associated with acetylcholine, soronin actually attached with this subunit. So soronin also attached with this cohesin, soronin, in this S phase. And in the S phase, what happens? DNA replication occurs. That means the chromosome becomes two sister chromatids. And during this phase, this cohesin actually gets the cohesive activity. By the help of soronine and COAT, it actually wrap the chromosome around the sister chromatid so that the sister chromatid actually adhere with each other. In the G2 phase, G1S and G2, in the G2 phase, two protein actually perform very important function. The name of the protein is MEIS322 and sugosine. These two protein actually recruit another important protein called PPA2. The full form of PPA2 is protein phosphatase A2. You can see here it is actually enzyme. These two protein recruit this enzyme to the centromeric region. This is the PPA2 recruit in this region, and also the cohesin are present throughout the chromosome surrounding the sister chromatid. But in the prophase to early anaphase, in this period, prophase to early anaphase, two molecules actually perform a very important role. The molecule name is called polokinase and another is called aurora B kinase. Must remember these two enzymes, it's very important. These two enzymes actually phosphorylate the cohesin all over the cell, all over the chromosome. And when the cohesin actually gets phosphorylated, it removes from the chromosome. There is a PPA2, so it will be removed from the chromosome. But as the name suggests, protein phosphatase, that means it is an enzyme that can remove the phosphate group. So the cohesin that are surrounding the centromere, they are not get phosphorylated, they don't remain phosphorylated, so they are not removed. The cohesin that is surrounding this centromere remains attached, and that helps the two sister chromatids to remain attached with each other. Here the first story ends and the second story starts. I will show you in this class. This is the first story, the cohesin. In the second story, that starts with centrosome. In the G1 phase, there is only one centrosome present in the cell. The centrosome contains two centrioles that is orthogonally located. From the centrosome, microtubules actually emanates or radiate. In the G2 phase, there are two centrosomes. The centrosome divides and there is two centrosomes. These divisions actually starts from the G1 to S phase transitions. And in this case also, polokinase helps in the separations. Okay. Now let me draw a cell, then you can understand. Let's suppose this is a cell, and think that this is the centrosome, two centrosome in different pole, and it actually radiate or emanate microtubules from different directions. I am showing only 3 so that you can understand. The two centrosomes radiate microtubules. The name differs. This type of centrosomes are called astral centrosome, astral microtubule, and the microtubule that attach with the chromosome, I am showing only one chromosome for the simplicity. The microtubule that attach with chromosome are called kinetochore microtubule. This microtubule is called kinetochore microtubule. Why it is called kinetochore microtubule? Because in the centrosome there is kinetochore, a complex content that actually attach with the microtubule, kinetochore microtubule. And those that do not attach with the chromosome are called interpolar microtubule. Okay. Now I have shown here that the chromosome is attached with the two microtubule from the two centrosome, but this is not only the situations. There can be 4 situations of the microtubule arrangement, and here from the second story starts. I will draw 4 chromosome with 2 sister chromatids. Let's suppose this is a sister chromatid, this is another sister chromatid, this is another sister chromatid, and this is the 4th sister chromatid, because I will give 4 examples of the microtubule rearrangement. This each and every chromosome or the sister chromatid have 2 kinetochore attached with 2 other. Now in the perfect situation, this is called amphitallic. That is the perfect situation, perfect arrangement of the microtubule, amphitallic arrangement. Okay, amphitallic attachment, you can say also amphitallic attachment. In these situations, both the kinetochore attach with microtubule from the 2 centrosome. Let's suppose this is a centrosome and this is a centrosome. 2 microtubule attached with this 2 different kinetochore. This is called amphitallic arrangement. This is the perfect one. But another thing can occur, that is called merotallic, in which one kinetochore attached with a microtubule and the same kinetochore attached with another microtubule from the other centrosome. Let's suppose this is a centrosome, this is actually a centrosome. One kinetochore attached with 2 different centrosome. This is called merotallic attachment. This is not good. And there is another way, that is called syntelic attachment, in which 2 different kinetochore attached with microtubule from the same centrosome. 2 different kinetochore attached with same centrosome. This is also not good. And another is called monotallic, in which one kinetochore attached with a single centrosome. Why I am telling that these are not the good situation? Because if you see here, this will, when the microtubule actually separate, helps in the separation of these 2 sister chromatids, they will not separate perfectly. Only these situations, the 2 sister chromatids get separated actually perfectly. So how cell sense these are not actually right conditions, because I am telling you these are not right conditions by looking at these situations arrangement. But how cell sense that these are not actually the right arrangement? There is a very important conditions actually cells can sense, that is the tension in this kinetochore region. In this region, cell can sense, actually molecules can sense the tension of these microtubules. When the microtubules attach with 2 different kinetochore from the 2 different centrosomes, they actually pull the 2 sister chromatids. And as I have mentioned in my first story, that in this region cohesins are only present, so this cohesin actually prevents this pulling, so they actually create these tensions. And when there is a tension in the kinetochore, there is a protein called NDC80, and in the center of the kinetochore, if I draw the kinetochore looks like this. In the outer portion of the kinetochore, there is a protein called NDC80, and the center of the kinetochore have aura B kinase. Aura B kinase. I have mentioned in my first story that aura B kinase phosphorylate the coenzymes and remove the coenzymes. Here also aura B kinase play an important role. How molecules sense that this arrangement is perfect? Here you can see NDC80 and aura B kinase present in the kinetochore. Aura in the middle and NDC in the outer kinetochore, outer part of the kinetochore. When there is enough tension, then this NDC80 actually gets separated from the aura B kinase, and now the aura B kinase cannot phosphorylate this NDC80 when there is perfect arrangement. You have to understand this. NDC80 present in the outer segment of the kinetochore. When this pooling occurred, this NDC80 actually separated or actually distant from this aura B kinase from the center of the kinetochore. Then this enzyme cannot phosphorylate this NDC80. Now in this situation, in this merotelic, syntelic and monotelic situations, this tension is not actually formed, because here the coenzymes cannot sense the pull strength from both these centrosome, because these are not goodly arrangement, rightly arranged. In that situation, the NDC80 or the kinetochore, outer kinetochore remains very close to the center kinetochore, and then the aura B can phosphorylate this NDC80, this molecule. And what happens when this aura B kinase phosphorylate this NDC80? This NDC80 actually lose the capability to attach with the microtubule. They lose the capability to attach with the microtubule, so microtubule cannot bind in that situations. And that's why all the chromosomes that actually not arranged, actually perfectly arranged, attached with these microtubules get a chance. So this attachment, this amphiteric attachment, because we know that there are so many times cell cycles occur during our body, but there is no problem at all in that situations, in these positions. All the chromosomes separates very right in positions. So how this occurs? This occurs by error and trial mechanism. The microtubule tried to attach with each and every way. Many times merotelic, syntelic and monotelic occurs, but cells can sense, and then another time the microtubule try. And when all the chromosomes, in case of humans 23 pairs occurs, when all the chromosomes arrange in this amphiteric attachment, only then this separation can occur. Now this is the end of this second story. Now I will show you the last story, that is how when these all chromosomes are actually in amphiteric attachment, when they are all in amphiteric attachment, how they are separated. Because now in that situations, in the anaphase, the chromosome, those are amphiteric in situation, that means attached with this microtubule from the both centrosome, okay, but they are actually attached with this cohesive molecule, and unless or until this cohesive molecules cannot be broken, they cannot be separated. So how this occur? Here comes the role of the mCDK, that is CDK1, M-phase CDK, and another important protein actually helps in this phase called anaphase promoting complex, APC by C. If you have not watched my previous video, in that previous video, first video, I have mentioned the name APC by C, and associate this protein APC by C actually associate with another protein called CDC20. Okay, so there are two proteins actually help in this process: CDK1, that is M-phase CDK, and these proteins. How they actually remove these coenzymes? The process is, there is an enzyme called separase. This is actually a protease enzyme that can cleave the coenzymes, but this separase also attach with a protein that actually do not help this separate to perform its function, that is called securin. So you can see here that the cells actually go or actually run by a very delicate mechanism, and each and every molecules are very connected with each other. You can see here separate is an enzyme, cannot perform its function when it is attached with securin. Okay, another thing is in the M phase, during this M phase, mitotic phase, these CDK molecules can phosphorylate this separate, and when this separate is phosphorylated and attached with securin, it is inactive in conformations. It is inactive. It cannot perform its function. Now the actually story begins from this anaphase, because we know that M phase means mitotic phase can be divided into prophase, metaphase, then the anaphase, and we know that in the anaphase two sister chromatids get separated. Okay, so in the anaphase this protein, this APC by C which associated with CDC20 actually gets activated. This protein actually first degrade this M1 CDK. So CDK1 is not there during the anaphase, so they cannot phosphorylate this separase. Another thing is this enzyme, actually this is actually ubiquitin ligase enzyme that actually ubiquitinates this securin and helps in the degradation of this only securin molecule by proteasomal degradation. Only then the separase is now free from this all burden and can perform its function by degrading a molecule that is present here called SCC1. When this SCC1 is cut, then the coenzyme cannot attach with this area, and then what happens? The chromosome get separated. Hope this video will help you. If you have any doubt, you can write me in the comment section. Thank you for watching, and always keep learning.